So hopefully no recording issues today. Last time we finished up muscle, and I wanted to see if there were any particular questions related to muscle. And then I really spent about 30 minutes, right, going over those first 9 or so sections. And when I went back, I realized that last semester I only spent 7 minutes doing that. So I gave you a greater, longer conversation about the review things than what I have done previously. But regardless, is there anything at all in the basics of the heart, basic anatomy, basic structures that I quickly went over, or anything you're concerned about from even lab, EKGs? That EKG conversation is going to make more sense as we go through this conversation today. It's going to start pulling together, I assure you, for all 6 of you. And those who are 5 of you, no, how many are there? 6 of you? Yeah, that are here. And but are there anything right now? Anything at all? Okay, keep pushing through this, and it'll all make sense. We got through, as I recall, the first 8 sections as a quick review last time, and that brings us to section 9 today, which is the beginning of the cardiac cycle. And this is going to look and sound a lot like the EKG conversation, okay, as we start talking about what's going on with the cardiac cycle. But we'll dive in a little bit deeper, but we'll also is that better, folks? We'll also understand what's going on with the EKG more completely. And we'll probably spend all of our day in this little section, okay? So the cardiac cycle. This is the time from R wave to R wave, right? From the complete cycle of the time that your heart is going to beat and then relax. The systole is the contractile phase, and the diastole or the diastolic is the relaxation phase. Now, typically, when one says systolic phase, they're referring to the ventricles, and only to the ventricles, because the ventricles are the ones that are doing all the work, aren't they? They're the thicker, more muscular chambers. So if someone just says in general, systole or systolic pressure, you're referring to the ventricles. But we will be discussing today atrial systole, right, when the atria are squeezing, and I'll distinguish those two. Okay, but typically if you just hear about systole and diastole, we're talking about the contractions. Now, the systole is when the blood is being pushed through the chambers, and then the relaxation period is when those chambers are relaxed, and when they're relaxed, they're still doing something. When they're relaxed, they're filling with blood. So we want to think about not only are we squeezing to push blood forward, but when we're relaxing, that chamber is refilling blood, or that chamber is being filled up with blood. So just a quick, easy enough visual. So relaxation phase. Now the heart spends more time in relaxation than it does in contraction. Okay, in the overall cycle of things, the heart spends more time relaxing than it does contracting. But we're going to see that it's a cycle and that it's very rhythmic, and we'll break down this whole contraction phase. Thank you.
What I do for some people I like.
Thank you. Okay, so the cardiac.
No nuts.
Okay, dark or milk? Dark. Okay, I'll buy 2. Okay, so the sequence of contraction. So we've got how does this whole thing going to work? We appreciate that the atria are going to contract first. They contract at the same time. Both the right and the left atria are going to contract at the same time. If they didn't, we'd be in a lot of trouble, and things would not work properly. And when the atria are contracting, we imagine that the pressure is increasing in the atria, and as a result, the valves open. Remember that the valves are not opening and closing like a cuckoo clock, right, on some sort of timer, but instead the valves only open when they're pushed open by adequate pressure by the chamber. So when the atria are squeezing, we're pushing blood through what 2 valves? On the right side, down through the tricuspid or the right atrioventricular valve. On the left side, we're breaking open the bicuspid, the mitral, or the left AV valve. So when the atria are squeezing, the ventricles are receiving that blood. right? And then the ventricles are going to squeeze. The ventricles are going to contract. That's going to be ventricular systole. Now, what's going to happen here? The blood is going to be pushed up and out. Remember that when the ventricles squeeze, that they squeeze not just as, like you're squeezing something, but they squeeze and they twist. They're pushing that blood up and out like they're wringing out a mop. And they squeeze and they twist, and that's going to push blood up and out what? Valves. From the right ventricle, we're going to go up and out the pulmonary semilunar valve into the pulmonary trunk. From the left side, we're going to push open and go out the aortic semilunar valve into the aorta. When the ventricles are busy squeezing, what's filling? Right, the atria are back to relaxation and they're refilling. Now, can you imagine, I'm going to show you this again in a moment, but do you remember that those nerves that go down through the interventricular septum and go around at the apex, those are the Purkinje fibers? And those Purkinje fibers not only go around the outside of the ventricular wall, but they also go to the papillary muscles. Can you picture those papillary muscles, those little nipple-shaped muscles on the inside wall of the ventricles? So what does that tell us? When the ventricles are squeezing, folks, yes, they're squeezing and they're twisting, but they're also causing the contraction of the papillary muscles. So when the papillary muscles are contracting, they're getting shorter, and what are they pulling on? The chordae tendineae. And what's that assuring? As the ventricle's squeezing, it's pulling down on the chordae tendineae, which is going to assure that the valves are kept closed. Because what valves are attached to the chordae tendineae? The atrioventricular valves. So when the ventricles are squeezing, you want blood to go up and out the semilunar valves. You don't want blood going back up in the atria, right? So when the papillary muscles shorten, it's going to pull down on those chordae tendineae, and that's going to cause the valve, the AV valves, to come down tight, right, and prevent leakage and backflow. Are we getting all this? Is it making sense anatomically? Okay. Now, typically, this whole thing lasts about 800 milliseconds. A fancy way of saying 0.8 seconds. And that was the average cardiac cycle time that I told you in lab. I said 0.8 seconds was the average cardiac cycle. That's the same as 800 milliseconds. So we see here just a picture of this cycle from relaxation to relaxation. First, you see in this visual, the atria are going to contract, and then the ventricles will contract. And this timing is critical to the overall normal functioning of the heart and to a normal EKG, as you're going to begin to put together these pieces. Another way of putting it would be in a cycle image like this. And these numbers you'll see again. But let's take a look. Atriosystole, here's the start. So the heart. Now, when this thing starts, when the atria begin to systole, when they begin to contract, what just fired? What causes the atria to contract? The firing of the SA node. Okay, so you can kind of put that right there. The SA node is at the start. It is firing. And the atria are going to contract. It takes about 100 milliseconds. And then the ventricles are going to contract. That's going to take about 370 milliseconds. Okay, that's a total of 370 milliseconds. So that means what? 270 milliseconds. You with me? Mm-hmm. So what do we notice? That the heart is in relaxation longer than it's in contraction. And that diagram shows you that in numbers. Okay? So the total about 800 milliseconds, 0.8 seconds. 100 milliseconds for the atria to contract, about 270 milliseconds for the ventricles to contract, and then the chambers in diastole or in rest longer than they are in contraction. So that's just an introduction to this. Now, here are those numbers again. Again, the average cardiac cycle is going to be every 0.8 seconds. That equals a heart rate of 75 beats per minute. How do we get that? 60 divided by 0.8, right? That is equal to 75 beats per minute. There's that atrial systole, the 100 milliseconds when the atria are contracting. And again, when the atria are contracting, right, they're filling up the relaxed ventricles with blood. And then when the atria are in diastole, that is the same as what? Atriodiastole is when the atria are resting, if you will, right? Okay. Then we go into ventricular systole, and we're going to break this into two parts, first and second phase. Ventricular systole, you are contracting the ventricles, and they're pushing on the AV valves. The AV valves are closed, but there isn't yet enough pressure to push open the semilunar valves. So this is going to be called isovolumetric contraction. Break that word down. Don't make it look like a big strange word. Iso, same. Volume, right? Isovolumetric contraction. There's no change in the volume of the ventricles. Before we even think about ventricular systole, I want you to imagine that the atria have already contracted, right? And the blood has already been pushed down into the ventricle. So we're starting, right? You're picturing a ventricle, and it's already filled with blood, and it's going to begin to squeeze. This isovolumetric time is when it's squeezing, but it hasn't squeezed, quote, hard enough to open up the semilunar valve. So this is isovolumetric because the volume is not changing. And then the second phase is where the pressure increases. The ventricle is beginning to squeeze harder. And we all know that Boyle, what did Boyle's law tell us? Boyle's law, and we'll talk about it again when we get to the respiratory system, but that was the dude that talked about volume and pressure, right? That they're inversely related. So if I squeeze the ventricle, the volume is getting smaller, which is going to increase the pressure. And so as I squeeze the heart, the fluid is building pressure, and that pressure is now going to push open the semilunar valve. Doesn't matter which one, right? If we're talking about the right side, we're pushing open the pulmonary semilunar. If we're on the left side, we're pushing open the aortic semilunar. At this point, the ventricles are going to start squeezing blood, right? It's going to squirt out and up into those chambers, or into those vessels. So this is called ventricular ejection. You are ejecting the fluid. It is leaving. Okay, so we have isovolumetric squeeze, isovolumetric contraction, no change in volume, followed by a greater squeeze where the blood does begin to eject from the ventricle. Then the ventricle begins to go into diastole, early diastole. The ventricle begins to relax, the pressure begins to drop, and the semilunar valves begin to close down. And then there's an isovolumetric relaxation. And this is where the semilunar valves are completely closed, the AV valves are still open, and there's no longer a change in the ventricular volume. And so this is considered isovolumetric relaxation. And the blood will be flowing into the atria. Do not get confused about aorta and atria, right? Just I know they look similar at first glance. So the atria, atrium plural. And then finally, there's a late ventricular diastole, and this is when all the chambers are relaxed, all the valves are open, and basically nothing's happening, right? The whole heart is in relaxation at this point. All the valves are open, everyone's relaxed. And the ventricles at this point will fill to about 70% of their maximum. Now, let's put a number on this. This ventricular diastole takes about 530 milliseconds. Now, this is all based upon that average 800 millisecond cycle. So we got 430 milliseconds to the end of the current cycle, and then 100 milliseconds more into the next before the ventricles will begin to contract again. So if you wanted to, this is the same diagram. The center of this circle is just like the one you have at the very top of your page, on the other page, and we've added the figures of the heart on this. So what we have here then, again, here's our beginning. Imagine the SA node is firing. We've got atrial systole, the first 100 milliseconds, and then we have atrial diastole, and that's all of this. So there's a lot of time where the atria are now relaxed. After that atrial systole, we go into ventricular systole. That's the other 270 milliseconds or so. And then this long diastole, which continues right into the next cycle until the ventricles once again begin to contract. So that's that totality of numbers I just gave you. So it's the pressure changes that occur in the ventricles, that occur in the atria, that is so important. You have to have enough pressure to open up the valves. Okay, like I said, the valves are not just opening and closing like a cuckoo clock on schedule, right? It's the pressure that's established by the heart. So if the heart cannot adequately squeeze, that means blood cannot adequately flow, right? So if the person has a damaged heart and the heart is not able to squeeze hard enough, it's possible that the person's heart isn't strong enough to even push open the valves. At this point, the heart is not really useful to the person. The valves require that increase in pressure, and then there's also a little bit of a recoil as they close. Now look at this image. This kind of puts everything together into one image. This is one of those images that I think you need to stare at with perhaps a full screen. Okay, so there's probably 2 or 3 images in this presentation, and this is one of them that I think would warrant your going into PowerPoint and printing this out in a larger size so you can scribble on it, or going to your book, this is 1910, and staring at it there. Because this puts it all together, but in a graphical representation. It's important that you kind of follow along the steps here. So across the top, we've got our atrial systole, our atrial diastole, right? What we've been looking at on the, sort of take that circle and make it a big long line. And then it also has across the top what's going on in the ventricles, or diastole and systolic cycles. You need to be able to recognize what's going on in each of these curves. So pressure is increasing on the side, and we've got time across the bottom. So number 1, the atria begin to contract. That's down here. Okay. And then number 2, those atria are going to begin to eject blood into the ventricles. Boy, that doesn't take very much pressure, does it? Okay, so the atria are not very muscular. Next week in lab, we'll be able to dissect a heart. You've seen a heart before. We won't spend much time on it, honestly, but I do want you to kind of see a heart once again, imagine the chambers, imagine the valves, imagine the flow of blood, and to once again appreciate the thickness of the ventricle, the thinness of the atria, to once again see how thin those vena cava are versus how thick the aorta is. So I want you to have that visual once again. But you see that the pressure that increased was not that much to get the valve, the AV valves to open. Number 3, atrial systole has ended. You don't see a lot of pressure, right? I mean, there's just a little blip here of pressure difference. But then the ventricles are going to begin to squeeze. And now you're going to see a huge increase in the pressure that's necessary to open up the valves. So we have to now start to squeeze. Now during this phase, all of this, that would be isovolumetric contraction. The heart is the ventricles are beginning to squeeze. The pressure is definitely increasing, but the aortic valve does not open up until here. So during that period from here up to here, that's isovolumetric contraction. The valve has not been pushed open. Then, here you see the valve is opened up. So now the aortic valve opens up. That's a bad color choice. Now the aortic valve opens up, and the pressure increases for a while. That's all part of that ventricular ejection. The blood is being ejected. Once that aortic valve opens up, blood is being ejected. And then the pressure will begin to subside. The aortic valve will close. And then everything coming down will now be what? Isovolumetric relaxation. So the valve is closed, and while the heart is relaxing, the volume is not changing. The valve has closed. And then we're back down to the bottom. Question?
I thought when they were relaxing, they were filling with blood.
They're relaxing, they're filling with blood, but the relaxation, the aortic valve is closed. The AV valve, look to here, follow with me. You jumped ahead of me, and I wasn't there yet. It's okay. At the bottom, that's where the AV valve opens.
Oh.
So then it would begin to refill. Okay, you're very good. So that's exactly where but you see that the aortic valve has to relax, be closed, and the ventricle has to relax. you don't want to fill a ventricle that's squeezing, right? You want to fill an open ventricle. So you want the ventricle to completely relax, then open the spigot, open the AV valve, and allow it to refill. So perfect. So there's that AV valve opening here at the bottom of this. And here, I didn't say it, but there's the AV valve closing.
So is part of it too with the atrium where it doesn't take a whole lot of...
Doesn't take a whole lot of pressure to open up.
Does it have to do with like gravity too?
Absolutely. Gravity certainly helps, right? The atria blood going down to the ventricles, because gravity is, you're perfectly right. So the squeeze is not significant, and gravity certainly helps that blood just to simply drop through and fill up the ventricle. Whereas the ventricle has to squeeze hard, not only to maintain blood pressure, but to get it up and out, because gravity is working definitely against the ventricle. Good. Okay, so this is one of those graphs. If it makes sense, fantastic. Follow those 8 steps along the right side and put this into a visual. I mean, I think this takes a visual recollection. And this image may or may not even show up on the exam. I might or may not. But you need to understand what's going on in this graph. Okay, either to understand it, to study from it, and or to label or know what's going on in different parts of this graph. Now, next week in lab, we'll also be listening to the heart. We'll be using a stethoscope. You'll be placing it on a partner's chest, and you'll be listening for the lub-dub, 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 right? Lub is characterized as S1. It's called S1, sound number 1. Dub is sound 2, or S2. Now, what is causing those sounds? Those heart sounds that you hear with the stethoscope are caused by the rhythmic closing of the valves. So lub is the closing of the AV valves, right? Both the right and the left AV valves, the tricuspid and the bicuspid, are closing. That causes a percussive sound that one can hear in the stethoscope. Then the dub is essentially when the semilunar valves close. So sound 1, AV valves, sound 2, semilunar valves are closing. Now, it's not just the percussive closing of the valves. It's also the backflow. Imagine when you have an open dam and then you close that dam, the water's going to come in and push up against what those newly closed doors. And so the sound is actually a combination of the percussive closing and sort of the sound of the blood pushing up against a newly closed valve. There are other faint sounds that are rarely heard in adults, S3 and S4. So I'm not going to really worry about them. Across the bottom, right, you can see this lub and then the dub. And then S3 and S4 are not very common. S3, if you hear it, is caused by the blood flowing into the ventricles, and S4, the atrial contraction. But boy, those are not loud sounds. So I'm not going to be worrying about S3 and S4 with you. There will be a figure coming up that will put those sounds on this chart. So go back 3 charts and tell me, based on what I just told you, where would S1 be if you were to plot it on this graph? Okay, where would S1 be? S1 was what? S1 was the, I'm going to draw a line across the bottom. That's my sound wave, right? And I'm looking for the closure of the AV valves. And that was right here, right? So S1 would be right about there, wouldn't it? Okay? That'd be S1 because that's where the AV valves are closing. And then the aortic valve and the pulmonary valve would close here, right? And that'd be S2. So if you're going to superimpose S1 and S2 onto this graph, that's where you would see them. Does that make sense? S1, S2. Another thing we'll be playing with next week in lab is cardiac output. This is an incredibly important concept. As the name suggests, it's the amount of output, how much blood is being pumped by the blood or by the heart with each minute. So it's a volume. It's a volume of blood that is pumped by the heart in each minute. That is the cardiac output. Cardiac output is defined as, cardiac output is defined as the heart rate, right, the number of beats per minute, times the stroke volume. Stroke volume is the amount of blood pushed out of the ventricle with each beat. Right? So you have so much blood with each pumping of the heart times the number of beats per minute will give you the total volume of blood pumped per minute. Very important equation, simple equation, but a very important one. Cardiac output equals heart rate times stroke volume. That's just shown here visually. Again, heart rate times stroke volume equals cardiac output. So next week we'll have a demonstration of this. Average cardiac output, 6 liters per minute, 6,000 milliliters per minute. That's going to be your average cardiac output. Where is that number coming from? An average heartbeat of 75 times a minute, right? We said that the average heart beats every 0.8 seconds, which is 75 beats per minute. And this is new for you, the average cardiac output, average stroke volume is 80. So in other words, every time your heart squeezes, your ventricles squeeze, you're putting 80 mils of blood into your circulation. Okay, so 80 mils every time, times 75 beats is going to give you 6,000 milliliters per minute. Now the body can adjust cardiac output. Now how could it adjust cardiac output? If you need more cardiac output, if you need more blood and therefore more oxygen to your tissues, maybe the bear is chasing you, what would you do? Increase your heart rate. Well, you know that happens, right? Catecholamines, you're going to be increasing epinephrine. It's going to increase the number of beats per minute. But have you ever heard of someone getting an EpiPen, like they're having an allergic response and they throw in those EpiPens in their thigh? Or a person who has an asthma attack and they're taking the albuterol? Not only does their heart rate go up, but they'll tell you it feels like their heart is coming out of their chest. Because not only will the heart beat faster, but the heart will squeeze harder. So if you need to maximally increase your cardiac output, you'll increase your heart rate, but your heart muscle will actually squeeze harder. And if it squeezes harder, what's going to increase? Stroke volume, right? So you can increase both. You can squeeze faster and squeeze harder, and you can drive this thing way, way up, okay? Stroke volume can almost be doubled. Heart rate can increase by 2.5 times. That can really increase the cardiac output when needing it. And again, we'll be playing with a little demonstration of cardiac output, stroke volume, and heart rate next week in lab. Now, you've already been introduced to the conduction system. You know about the SA node and the AV node, and I'm going to just kind of go through that quickly with you. This conduction system is the specialized electrical activity of the heart. This is what's going to initiate the heartbeat. Now, we know from the autonomic nervous system conversation that sympathetic stimulation increases your heart rate, and that parasympathetic can decrease your heart rate, but they don't initiate it, right? They influence it. So your heart initiates its own heartbeat, but the sympathetic and parasympathetic nervous system branches can influence that. And we'll also see that there's hormonal effects on the heart as well. Okay? So we say the heart is autorhythmic. It expresses or has autorhythmicity. The SA node, you know where it is. What chamber are we in? Right atrium, kind of on the superior side of it, right? The model shows it sort of as the superior vena cava drops into the right atrium. It's rather high in the right atrium. These cells, I mentioned this in lab, do not look differently under the microscope. You cannot look at a patch of the right atrium and say, that is the patch of cells that are the SA node. They behave physiologically different. They have a depolarization that keeps happening, right? That keeps causing an action potential, but you cannot physically tell them apart by visual appearance. And then we would say the internodal pathway is what? Internodal, between the nodes. So the internodal pathway is the path that the signal goes from the SA node to the AV node. Right? Internodal pathway. Then, once the signal gets to the AV node, we know where that is, right? It tells us it's at the atrioventricular junction. It's at the bottom of the right atrium. Then there are other pacemaker cells there. Now, this is important. If the SA node is damaged, in other words, if the SA node is not firing at all, the AV node will still fire. But the AV node, those cells are different than the SA node cells. They will only fire at 40 to 60 beats per minute. Now, that's enough to sustain life. What would you see on an EKG if the SA node was damaged? I'd see QRSs, right? But I wouldn't see any P waves, right? So I would see no P waves, I'd just see a bunch of QRSs. And the rate would be a lot slower, right? Only 40 to 60 beats per minute, because that's how often the AV node will fire on its own. The SA node will fire about 80 times a minute, right? Well, 75, right? What's our normal heart rate? 75. So the SA node is what's setting that, 75 beats per minute. 75 to 80, somewhere in that range, is how often the SA node will, on its own, fire. But if it stops firing, the AV node will still be a backup generator. It will still back up and fire 40 to 60 times per minute. About the fastest you can go is about 230, okay? About 230 beats per minute, maximum normal heart rate. Any higher than that, there's something very significantly going wrong, okay? So the younger you are, the higher your heart rate can be during exercise. And if you look at those tables, about 230 is about as high as you would see. And as you get older, right, that maximum heart rate comes down because the heart can only take so much. If someone has over 230, they're either being stimulated by some sort of upper drug of some sort, or there's some significant damage to their conduction system for a long period of time. When the AV node fires, when it receives the signal from the SA node, the AV node then is stimulated and it fires. That signal is going to start traveling down the AV bundle. The AV bundle then turns into the bundle branches. The bundle branches are going through the interventricular septum, and they're going to bring that signal down to the heart's apex and bring it around. Now, the left branch is larger than the right branch. That makes sense because the left ventricle is a larger muscle mass and so requires more innervation. And then the dead end of this is down at the Purkinje fibers. These are named after a person who described them, so they're always going to be capitalized, and it is a strange name. Don't blame me, right? Purkinje, but it does sound and spell the same way. And these fibers are going to head down, and these are large diameter, and they conduct very, very quickly. And they're the last little bit of the conduction system. And as you've seen in the model and as you'll see in cartoons, these go around the apex, along the outer ventricular wall, but also go right up into the papillary muscles. Okay, and they're going to be pulling on those papillary muscles during contraction. So on the hearts, you should be able to see these 5 or 6 things. On the models, you should be able to see them. On the lab practical, the next lab practical will not be a station exam. It'll be a sit on your butt and answer questions exam, and there'll probably be pictures of the heart model. But you need to recognize those 5 parts. And there'll be pictures of blood cells, and there'll be pictures of EKGs that you'll be analyzing on paper. I kind of wish here the nerves were shown in yellow, right? Because we're so accustomed to yellow being our nervous system color, but here they're shown in purple. This just walks you through this, and I think we're getting there. I think based upon lab, students are getting this. So let's take a look at this. I know this is small in your slides, but Action potential is generated at the SA node, right? The cells reach threshold, they're going to fire. That's going to cause the depolarization throughout the atria. That signal is going to spread quickly because of intercalated discs, and that's going to begin the contraction of the atria. What are we going to see on the EKG? P wave, right? The P wave is going to begin as the atria begin to contract. So it's showing that here. Here's the the SA node on this image, and we're going to start spreading. That's shown in purple. Spread, 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 and we're moving toward the AV node. Okay, we're moving toward the AV node. How long does this take? How long did we say this takes? Go back to the atrial systole. Was that 100 milliseconds? Right, about 100 milliseconds, just kind of get your time. It only takes about 50 milliseconds for it to get there. But here's what's interesting. There's a delay. When the SA node reaches the AV node, there's a short delay. It takes a little while for the AV node to get activated. And it's during this delay that the atria contraction will continue. And then once we have that little delay, then the signal runs down the bundle branches to the Purkinje fibers very, very quickly. Now that little delay, that little hiccup, that little 100 millisecond delay is very, very important for the heart to be working normally. If there wasn't a delay and the signal went from the SA node to the AV node and then straight down the Purkinje fibers, what would be the problem? Think about this. If there wasn't a delay and the SA node got to the AV node and the AV node just took that signal and flew around the Purkinje fibers and then the ventricles started to squeeze, what would you have not given the heart enough time to do? The atria are squeezing, and if the ventricles started to squeeze immediately, not enough time for blood to get into the ventricles. You're right. Not enough fill time. So that 100 millisecond, that tenth of a second delay in the signal transferring to the AV nodes is critical, because if it was too short or shorter, there wouldn't be adequate time for the ventricles to fill with blood. What good is it for the ventricles to squeeze if there's not enough blood in them? What would be decreased if the fill time was decreased? If there wasn't enough time for the blood to fill the ventricles in that cardiac output equation, what would be damaged or deficit? Stroke volume. So if there isn't enough blood in the ventricle when it squeezes, then your stroke volume will be way down. If your stroke volume's down, so too will your cardiac output be down. And if you don't have adequate cardiac output, you're dying, right? Your brain's not getting the oxygen it needs, the tissues aren't getting the oxygen that it needs. Yes, yes, starting to come together. It really is a beautiful story. It's complex, there's a lot of stuff going on, but it really does make sense when we start thinking about it. Well, let's talk about what happens in the cardiac muscle cell. Now, this is also, I think, largely review. You've seen skeletal muscle mostly, and you've seen what action potentials look like in skeletal muscle. We talked about them just last time, right? What's different about an action potential in skeletal muscle between that and a neuron? Was there anything? Wasn't the resting membrane potential still negative 70? The still was up to positive 30. The time lapse was the same. It was exactly the same stinking looking action potential, wasn't it? But what we're going to see is that the action potential in a cardiac muscle cell is going to be different. So to review, in a skeletal muscle cell, right, your biceps brachii, the action potential is very brief. That little twitch, right, is very short. The sarcoplasmic reticulum was the storage area for the calcium, and there was a very short refractory period. Can skeletal muscle reach tetanus? Right? Tetanus was, I was describing STEPI and then summation and then impartial tetanus and then full tetanus. And that was all a conversation about skeletal muscle. So a skeletal muscle can reach tetanus. That is, it's going to be so tight that it cannot do anything else. Would not be a good idea for the heart to go there, would it? So we're going to see that the heart cannot go into tetanus. When a person dies of lockjaw, what do they die of? Respiratory. Their diaphragm, skeletal muscle, locks and doesn't allow. But you don't hear about the heart being the problem, okay? Because the cardiac muscle is uniquely protected from going into tetanus, as we'll see in a moment. The diaphragm goes into tetanus. People with lockjaw die of respiratory failure. They don't die of the heart going into tetanus, okay? So let's figure this out. This is what happens in skeletal muscle. We've already seen this. You can sleep for just a couple more seconds, right? So we've got a very quick action potential up and down, and threshold is reached. Same old story. We know this story. Now, there's a little bit of a latency period, isn't there? What did latency mean? From the time the stimulation occurs, right? There's a little bit of a short latency before it begins to contract, just a couple milliseconds, and then it goes up and comes back down. We got that. Okay, that is skeletal muscle. Cardiac muscle, though, is different. Cardiac muscle is going to have a much longer action potential. It's going to take a lot longer. Calcium ions are going to enter over a longer period of time. Calcium, again, is very, very important here. And the contraction can last an average of 250 milliseconds. The refractory period also continues long into relaxation. So this means that tetanus, right, cannot occur. Tetanus cannot occur, will not occur in the heart. Otherwise, we'd be in trouble, right? A heart in tetany could not be pumping blood. A heart that was just, sitting there squeezing would be useless. So let's take a look at a cardiac action potential. And we notice that this does not look like an action potential that I've seen before. Typically, they go up and down really, really fast. So we have a resting membrane potential. It goes up fast. That's the same. But when it gets to the top, it tends to kind of hang there, and then there's a long plateau before it crashes down and repolarizes. It's this length of the plateau that is going to keep the heart from being able to go into tetany. Okay. And you also see that there's this kind of long, slow contraction. And I just told you that the average contraction was what? This is 250 milliseconds. So what does that recognize? 250 milliseconds, go back a slide, was what I told you. Oops. 250 milliseconds was what I told you how long the typical cardiac muscle cell is contracting. And you look at this and you see that 250 milliseconds right about here. And that brings you to the end of the relative refractory period. But see, there's an absolute refractory period that's in blue. And what does absolute refractory period mean to you? It cannot start no matter what, and that's 200 milliseconds. Okay? There's no way the heart's going to beat again faster than that. Now, that would still be way too fast, wouldn't it? If the heart was beating every 200 milliseconds, what would the heart rate be? Really high. Good. 200 milliseconds, there'd be 5 of those, right, in a second? Right? So what would be their 5 times 60? 300? Right? In other words, there's no way the heart can go over 300. Right? Do we see why? There's no way the heart can go over 300 beats a minute. Physically can't because of this safety switch, this long action potential and this absolute refractory period. And you would never want to sustain over 230 anyway, right? Like I said, over 230, there's something wrong. Either a person's on simulation drugs or something, or there's an electrical conduction problem. Now, we know the stages of this. And Martini kind of goes through this story again with you, because you can imagine in different schools and different scenarios, you may be learning about the heart before you learn about action potentials. So some people would learn about this in the heart before they would go on and learn about the nervous system. So they have to kind of go through this whole thing. But you know the three stages. There's a rapid depolarization, up really fast, really up. That's the same. All related to sodium, all related to voltage-gated sodium channels. And this is all about the same influx of sodium. These are very, very fast sodium. They remain open for just a short while. But then here's what's unique. There's that long plateau. When you get up to about 0. Now what's happening here is that those fast sodium channels close, just like they did before. But there are voltage-gated calcium channels that stay open. And this keeps that plateau up there. And these are called slow calcium channels because they're slow to close. They open slowly, they remain open for a long time. And that calcium, let's think about it, that calcium is positively charged, isn't it? So that keeps that membrane potential really high, and it allows for that long plateau that one sees. And then there is repolarization. Those slow calcium channels will eventually close, and these potassium gates are also kind of sluggish, and they will finally open up. And it's the potassium gates, as we know, that does what? Right, helps to restore the membrane potential. So then we get a more rapid repolarization, but not until there's that long plateau.
No hyperpolarization?
Little tiny bit, but not a lot. You don't see a lot of hyperpolarization, so that's a good point. And that's because it's so slow. In a normal action potential, it was that rapid sodium and then rather rapid potassium gates that opened, and they tended to overshoot and cause the hyperpolarization. Because these are slower, there's no tendency to overshoot, so you don't see the hyperpolarizing event here. So here is that cardiac muscle cell action potential, and you see it now with time. There's that 250 milliseconds, representing the end of the refractory period. Here's the absolute refractory period at 200 milliseconds, saying that the heart could never, never, never beat more than 300 times beats per minute, right? Never go above 300 beats per minute based upon that plateau. So again, this is why the heart can never go into tetany. You'll never get summation either, would you? Because summation required what? I mean, if you think about those graphs, can you picture those graphs? Mm-hmm. Summation meant that there wasn't enough time to come down before the next stimulation. And so you won't see summation, you won't see tetany in cardiac muscle. Now, what is it that causes the heart to beat? We keep saying it's autorhythmic and that the heart has an SA node. What is it about the SA node? What is it about those cells that allows them to cause the action potential? And it turns out that those cells in the SA node and those cells in the AV node do not maintain a resting membrane potential. So when you think about a resting membrane potential, you think, oh, a neuron just sits there and sits there and sits there and sits there at -70 until it's stimulated. But it has a stable resting membrane potential. These cells in the SA node do not have a stable resting membrane potential. So they're always drifting up toward threshold. Always, always, always. And the SA nodes simply reach threshold before the AV nodes. I told you before, the SA node fires about 80 to 100 times a minute. Now wait a second, I just told you a moment ago our heart usually only beats on average 80. So we know this story, but maybe we don't remember it. The SA node would on its own fire 80, usually closer to 100 times per minute. But the vagus nerve is always influencing the heart rate. Remember the vagus nerve, cranial nerve number 10? And there's what's called vagal tone. The vagus nerve is part of what? Sympathetic or parasympathetic? Do you remember what were the 4 parasympathetic cranial nerves? 3, 7, 9, 10. the vagus nerve is always sending a parasympathetic signal to the heart saying, cool it just a little bit. So while the SA node would want to fire at 100, it usually only fires around 75 to 80. Why? Because the vagus nerve is always influencing the heart. If a patient had their vagus nerve cut, damage to their vagus, their heart rate would necessarily go back up to 100 beats per minute at rest. Okay? So if the vagus nerve were damaged, you would lose that vagal tone. You would lose that parasympathetic brake on the heart. And that person's rate would go up to the intrinsic rate, up to the rate of about 100 beats per minute. Now, because the SA node is firing 80 times a minute, the AV node only fired how often? 40 to 50. So that means that the SA node is setting the pace. As long as it's working, the AV node would never fire on its own, would it? Okay, so you always have the SA node telling the AV node when to beat. It's when that SA node stops firing that the AV node would have the opportunity to fire on its own as a backup generator at 40 to 50 times a minute.
And when the SA node is working, the AV node is the same amount?
When the SA node is working, the AV node meets that rate, right?
Okay.
Because the time to go from the SA node to the AV node is really quick, and it tells the AV node to go, okay?
It's only slower when the SA.
Yeah, so the AV node's only going to fire as a backup, if you will, if the SA node did not fire. Okay. So here is what this looks like. This is basically the membrane potential of the SA node cells. And what do you see here? They don't have a rest, right? There's no, in other words, if I were looking at a neuron, I would see a long resting membrane potential. And then there'd be some sort of stimulation, and then you would see it go toward threshold. These cells have a leaky channel, so they're always, always, always creeping up. So they creep up. Now look what their resting is. Their resting's around -60. They creep up, they reach threshold, they have their action potential, they come back down, but then they just immediately start going back up again. Look at the timing of this, about every 0.8 seconds. Right, so about every 0.8 seconds, these cells are going to reach threshold, do their action potential, and return, and then return. Make sense? This is what's driving the SA node.
So instead of that hill that goes up and comes back down, this is what it looks like?
Yeah, this is what their action potential would look like. Now, I just spoiled the story. Here's the information if you didn't catch it all. The parasympathetic system is influencing the SA node. Okay. Parasympathetic, I just told you it was a vagus nerve, but what's happening is that there's a binding of acetylcholine from parasympathetic neurons. And you, for your purpose, can think the vagus nerve, right? This is the vagus nerve coming in. And that's going to open up potassium channels, and that's going to slow the rate of the spontaneous depolarization. So again, what the parasympathetic system is doing is putting a brake, a light brake, on the depolarization of the SA node. So look at this. Okay. So what's happening is that the parasympathetic stimulation is actually causing a slowing down. If you look back, I wish these were side by side. If you look back, what's happening is that this is causing this period here to be longer, right? It's slowing it down just a little bit so that it reaches at 0.8. Now, the last one just said 0.8, but imagine it would be going faster if it wasn't for the vagus nerve, if it wasn't for the parasympathetic stimulation. Now, sympathetic can also influence the heart rate. This is going to usually drive up the rate, isn't it? And this is going to involve binding of norepinephrine. So when the bear is chasing you, right, your heart rate goes up, and the molecules that are released by your adrenal medulla are what? Epinephrine, norepinephrine, right? Those are your molecules released when the bear is chasing you. And those that norepinephrine is going to bind to a beta-1 receptor. I know we didn't spend a lot of time there when we talked about the hormones and things, but that's going to bind to beta-1 receptors, and that leads to the opening of ion channels. Well, guess what? If you're opening up ion channels faster, then you're going to reach depolarization faster. That's going to shorten, not only cause you to reach depolarization faster, but it also shortens the time for repolarization. As a result, the heart rate increases. So epinephrine, norepinephrine, catecholamines are going to open up sodium channels, cause the depolarization to happen more quickly, quicken the repolarization, and as a result, shorten the cardiac cycle time. So we can see here now, under sympathetic stimulation, look where the 0.8 is. Normally, your heart would have only beaten once, right? And we see now it's beating faster. So that's what this is trying to show you. That in the same 0.8 seconds, we've gone through more than one cycle. We've actually had faster heart rate. I'm more faster. How's that for my grammar? More faster. Oh, this is where Stacy's going to glare at me.
Yeah.
Are we all ready? Stacy's going to glare at me. I'm ready. Maybe Renee will too. Just look at the reflection off my eyes. You'll see it. It'll be very bright. Resting heart rate, what we say, average, 75 beats per minute. And I told you in lab the average was what? 50 to 100. Why did I say 50 to 100? Because, well, it's easier, yes. And I told you that in lab, just to be straight up here, I told you in lab that I said 50 to 100 was average, because in the lab it said that a rate of 0.6, every 0.6 seconds was a rate of 100, down to 1.2, right? So if your heart beats every 1.2 seconds, that's a heart rate of 50, and if your heart rate is every 0.6 seconds, that's a rate of 100. And I even had you change in your arrhythmias from 59 to 49, right? Just to make it consistent. Martini's telling us that normal is 60 to 100, and I'm okay with that. So that means I need to be very clear on my test, right? So I'm not going to say a heart rate less than 60 equals. I would say a heart rate less than 50 is what? Just to be consistent and safe. So less than 50 we're going to call bradycardia, okay? And less than, over 100, that's consistent. The glaring just stopped. Over 100 is tachycardia. And the only reason I said that about Stacy is because she noticed, and appropriate so, that in lecture I said that lymphocytes were between 20 and 40%. And in lab, I said 25 to 33. And so she noticed that and let me know it, lovingly. Okay? So there are going to be some differences, okay, here and there. And I'll make sure that the examples on the test are grossly wrong, that they're going to include both of what I said in lecture or lab. So if you're looking at a normal heart rate, again, here Martini is telling us 60 to 100. Anything above that, tachycardia. Anything less than that, bradycardia. Now, let's think about this for a second, because this drives some students into a mental cartwheel. Tachycardia is a more rapid heartbeat. That means that the heart cycle is happening more frequently, right? So here, your heart rate would be 0.6 seconds or shorter, right, if you're going more tachycardic. And as you're going to bradycardic, that means your heart rate is more often than 1.2 seconds or so, right? 1.2 seconds would be 50, but somewhere in that range. Actually, 60 beats a minute would be what? This is easy math. Every second, right? So if your heart's beating less frequently than every second, Martini tells you have bradycardia. Now, do some people walk around with really low heart rates? Yeah. Athletes especially will walk around with naturally low. Now, why would an athlete have a lower resting heart rate? Potentially.
It's always working at a faster rate.
I'm going to say this. I'm probably a little bit ahead of myself. The body needs to maintain this cardiac output. For your body to work well, it has to maintain cardiac output. And we know that cardiac output is equal to heart rate times stroke volume. A very fit heart is very efficient and has a very good stroke volume. So if your stroke volume is very efficient and high, that means your heart rate would not have to be as often to maintain the same cardiac output. So a person who's less fit, and I shouldn't say just their body, but their heart, if their heart is less fit, that means it's not squeezing as hard, right? If it's not squeezing as hard, that means the stroke volume will go down. How would that person's heart naturally have to accommodate to maintain cardiac output? Their heart rate would have to go up. Now with age, heart rate does go up. Right? With age, naturally, heart rate goes up. Why? The body's trying to maintain cardiac output equally. Your brain still needs as much oxygen when you're 75 as it does when you're 25. So since stroke volume will naturally go down with age, because as the heart gets older, more tired, right, it just isn't as efficient, if you want to think of it that way. The heart will have to compensate with driving up the heart rate to maintain and keep cardiac output the same.
So when you're older, your stroke volume is lower?
When you're older, your stroke volume would go down. And we'll get to that in a few minutes, but the heart becomes less elastic. It becomes, tougher, right? Veal's more tender than beef. Right? So, another way of putting it, right? So old people's muscles just are a little chewier, okay? So the heart does not. So the heart, as it gets older, does not squeeze as easily and is a little bit less, and that means the stroke volume goes down, and the body has to drive the heart rate up to accommodate. Okay, so as a natural consequence, forget the veal, the stroke volume does go down, the heart rate does go up. Because we need to maintain cardiac output the same pretty much throughout life. Now, what is initiating this? You've heard me say that the heart is autorhythmic. But there is a backup plan here. That is the medulla oblongata. Your cardiac centers are in the medulla oblongata. There is a part of your medulla called the cardioinhibitory center. And this is what's controlling your parasympathetic neurons, and this is what's decreasing your heart rate. I just told you the vagus, right, was in charge of this. So the vagus nerve is being influenced by this cardioinhibitory center. And what it says is that signals coming from the medulla are traveling through the vagus nerve as part of the parasympathetic nervous system, and they are synapsing on the cardiac plexus. The cardiac plexus is just a fancy way of saying all the nerves that go to the heart. Right, the group of nerves that go to the heart, the cluster, the bundle of nerves that go to the heart is the cardiac plexus. And what that does is it's going to slow down the intrinsic rate. So, but what's intrinsic? What's intrinsic mean to you? If you are intrinsically motivated, you are motivated from within. It's your natural tendency to do something. So what is the intrinsic heart rate? 80 to 100. Right? That's how fast the SA node would want to fire, but it's influenced by the vagus nerve through the parasympathetic nervous system and causes it to cool down to 75 to 80 or so as an average. So that's that vagal tone. Vagal tone. So here's a visual of this. Okay. So here we are. We're way up in the, you can't appreciate that, but we're up in the medulla oblongata, part of the spinal cord where it comes up into the brainstem. And here's the vagus nerve coming down. And the vagus nerve comes down and talks to the heart and says, cool it, right? And how's it going to do that? It slowed down the ability to reach threshold, didn't it? It lengthened the time to get to threshold. Now, sympathetic, if there was a sympathetic signal coming down, what would it cause the heart to do? That would increase the heart rate, wouldn't it? Okay. But parasympathetic is going to drive the heart rate down. So there's a visual, vagus nerve coming down. More about this in a moment. There is also a cardioacceleratory center. So there is a part of the medulla that can increase your heart rate. Now, under what situations does your heart rate increase? You're scared. You're nervous. The bear is chasing you. Something sympathetic. Okay. Caffeine. right, can have an effect as well, can't it? Okay, so that can do it as well. I keep spoiling this. So I've already told you that throughout your life, cardiac output needs to stay about the same. CO equals HR times SV. As you get older, heart rate goes up. Why? Because stroke volume is going down. Okay, so we need to maintain. And what should our normal cardiac output be? 6,000, right? So just as a guess, just as a number to keep in our head. So about 6,000. So we need to keep this going because our tissues must continue to be perfused. The word here is perfused. You need to have all of your tissues perfused. It's a way of saying, are they getting enough oxygen? If your perfusion goes down, then that means your heart's not pumping enough blood to that particular organ. Now, what organs are especially finicky about receiving blood? The brain, right, must receive blood constantly. What's another big one? We've talked about it even a little bit. What other organ is kind of measuring your blood pressure and making hormonal? Yeah, the kidneys, right? So if the kidneys are not receiving sufficient blood pressure, how did they respond, Matt? What did the kidneys do when they got mad about not having enough blood pressure? They put out renin, right? So we've already seen that the brain is upset. They don't, that's not good. The kidneys also are looking for sufficient blood pressure because without sufficient blood pressure to the kidneys, they don't work. They try to stop that from happening by kicking out renin and increasing the blood pressure. And your muscles also require quite a bit of perfusion, not at rest, but of course at exercise. Now we know the heart's a two pump organ side by side. And we always talk about the left ventricle because the left ventricle is the one that's pumping blood to your brain and to all of your tissues. But the right ventricle must be pumping at the same rate. Right? I know I asked this question last semester. Which side of the heart, left or right, is pumping more blood? And I usually get all kinds of answers until we think about it. Because both sides have to be pumping the same volume. Because it's a closed system, right? You've only got about 6 liters of blood. And that 6 liters of blood has to go somewhere. And if you are pumping more from the right than you were the left, then there'll be a backup or an overflow someplace. So the right and the left ventricles, even though they're different sizes and even though they are different strengths, are pumping the same volume of blood. They're pumping under different pressures, right? The right ventricle does not have to pump as forcefully to get the blood to the heart, to the lungs. But they're pumping the same volume. Otherwise, right, do you agree? There'd be a big backup or buildup of or overflow of blood somewhere. So both sides are working the same. So we think of the double pump, but they have to be working together. Now here's a couple more volume things. And we're back into the heart, and we're thinking about stroke volume. And what is stroke volume again? It's the amount of blood pushed out of the ventricle. Now here we're talking about just the left ventricle, okay, into the aorta. So stroke volume is the amount of blood pushed from the left ventricle into the aorta with each and every stroke. Now it's the same volume going over on the right side, going to the lungs, right? But we're only going to be talking about or thinking about right now the amount of blood going to the body, to the systemic circuit as we discuss this. At the beginning of the cardiac cycle, when the ventricle is at rest, the amount of blood sitting in the ventricle is called the end diastolic volume, okay? At the end of diastole, I think I may have said that wrong. At the end of diastole, at the end of relaxation, the amount of blood in the ventricle is called the end diastolic volume, EDV. EDV. We'll put this together in a second. As you begin to squeeze the blood out, and now you forcefully eject, right? Ventricular ejection, you force blood out. What's left after you've squeezed is called the ESV, end systolic volume. Now, what did I tell you the average stroke volume was. Remember what I said? Cardiac output was heart rate times stroke volume. Average heart rate we know is 75. Stroke volume was average of 80. And that gave us the 6,000, didn't it? Okay, you with me? Where does this stroke volume come from? Stroke volume is EDV minus ESV. The amount of blood that started in the ventricle minus the amount that was left, that was pushed out. And that's going to equal, on average, 80 milliliters. 80 milliliters. So on average, this is going to be 80 milliliters. So every time the ventricle squeezes, it's pushing on average 80 milliliters into the next place. 80, 80, 80, 80. So we start, and if you could measure the volume in this, what's happening right now? You see blood going down through the atrioventricular valves. So at this point, it's ventricular diastole, and the ventricles are doing what? Well, they're relaxing, and they are filling, right? They're filling with blood. And at the end of that filling period, at the end of that rest, at the end of that diastole, the amount of blood that's in here is now called the EDV, right? That's the EDV. And then the ventricle is going to squeeze, right? It's going to pump. And as it squeezes, it's going to push blood out. At the end of that squeezing, the amount of blood left in the left ventricle is called the ESV. In other words, the heart does not squeeze out all the blood. A lot of it, but not all of it. And the amount of blood each time that it pushes out is going to be about 80 milliliters. So you're going to take the end diastolic volume, and you're going to subtract the ESV, the end systolic volume, to get your stroke volume.
So in a weird way, this is kind of like a car.
In a
gas
weird way,
tank.
this is like a car gas tank.
Like, because you don't use all of the gas.
We hope we don't run out of gas, right? So we don't use it all. And as you're running along the highway, it's like your gas tank is squeezing, right?
Like in a weird way.
Yeah, in a weird way, but it's okay. That weird way works, right? It works. I can see the analogy. It's not perfect, but I see it, okay? I see it. Because I don't think the gas tank's squeezing too much, but that's okay. It's definitely emptying, though, isn't it? Now, what's going to influence stroke volume? So let's think about this. I'm telling you that the body must keep cardiac output essentially the same. When you're exercising, your cardiac output can go way up. But there's a minimum at rest to maintain homeostasis. So what can influence your stroke volume? Well, let's first of all think about what can influence the end diastolic volume. Okay, what can influence the end diastolic volume? Well, the number one thing would be, first of all, what is EDV? Can you give me a picture? Can you describe it to me? The end diastolic volume is the amount of blood sitting in the heart sitting in the ventricles when the atria are done squeezing and before the ventricle begins to squeeze, right? That's the EDV. It's the amount of blood just sitting there waiting at the end of the diastole, at the end of the relaxation. Ventricular. This is all about ventricles. So EDV is the amount of volume of blood in the ventricle at the end of its relaxation. It's about to start to squeeze. So the ventricle's been sitting there waiting. It's resting. While it's been resting, it's been filling from blood from the atria. So the EDV is the amount of blood sitting in the ventricle before it squeezes at the end of its relaxation. Now, what's going to have influence that? Number one, venous return. If there isn't good venous blood coming back to the heart, do you agree that you might be influencing in a negative way, or I suppose a positive way, your EDV? More blood, less blood, right, would be influencing the end diastolic volume. Now, that could be influenced by blood volume. If you have a reduced blood volume, then you won't have as much blood coming into your heart, will you? If you're hemorrhaging out, if you're losing blood somewhere, then that means not as much blood's coming back. That's certainly going to drop your EDV. Also, muscular activity. If you're exercising, it's going to increase your EDV. Why? Because with skeletal muscle pump, as you exercise, as you breathe deeper, more blood comes back more rapidly. So your venous return increases. That would increase your EDV. And what if you had the rate of blood flow through your peripheral tissues? Okay, and that could be either way as far as increasing or decreasing the amount of blood coming back to your heart. Now, what's the problem here? EDV is also largely influenced by your filling time. How much time your atria have to refill the ventricles. What did I already say was critical? What is it that keeps your filling time adequately long? The amount of time it takes for the time for SA to AV node, but also remember there was a 100 millisecond hiccup at the AV node. There was a delay. There's a little hiccup in the switch, right? So there's a 100 milliseconds delay from the time the signal gets to the AV node until it picks up that signal and continues it. The latency period so far has been the time from the time stimulation occurs until the muscle begins to contract. That'd be latency. This is simply it's the AV delay. Okay, we'll hold it for there right now. Now, again, during that AV delay, what's happening? You're giving more time for blood to fill down into the ventricles. Okay, so that's the duration of the ventricular diastole. What if you had a slow heart rate? Slow heart rate. A fine-tuned athlete, right? Well, or it could be bad too, right? But a slower heart rate. That means your heart has more time in relaxation, which means there's more time to fill, isn't there? So if you have more EDV, your stroke volume would go up. If your stroke volume goes up, then your heart rate could go down while maintaining your cardiac output. Say that again. If you have an increased EDV, that suggests that you would also have an increased stroke volume. Increased stroke volume would allow the heart not to have to beat as often, lower the heart rate in order to maintain cardiac output. But what if there's an increased heart rate? Your heart's beating faster. It actually is going to decrease your fill time and decrease the amount of blood sitting in your ventricles, and that would decrease your stroke volume and necessarily increase your heart rate in order to maintain your stroke, your cardiac output. Okay, so we'll be dealing with this next week in lab. A couple more things, and then I'm going to take a quick break because you guys need it. Another thing that's going to influence stroke volume, preload. Now, this is a term that doesn't quite mean what it sounds like. Preload, when I hear the word preload, and maybe when you hear the preload, you're thinking, oh, it's just the amount of blood that's loaded in the heart before it begins to contract. That's what preload means to me. But preload, and it's not wrong, but it's not completely correct. Preload actually refers to the amount of myocardial stretching. How much stretching the heart muscle is having to do. Now, obviously, the more volume there is, the more it's going to stretch. So it's kind of similar to what I first said. Preload is how much blood is there. But from a physiological standpoint, it's not how much blood is there, it's how much the muscle is stretching. The heart, as you know, is stretching and recoiling and contracting all the time. Now, the greater the amount of blood in the ventricle, the greater the amount of stretching, therefore, the greater the preload. Now, who first described this were two guys, Frank and Starling. And so they're named together, the Frank-Starling law of the heart. And basically what the Frank-Starling law of the heart says is that the more you stretch the heart the harder it will respond back. So if you increase the amount of blood in the ventricle and you stretch it more, the heart muscles will respond by squeezing more forcefully to get it all out. In other words, the heart will do what it needs to do to keep the body going. So if you put more blood in the ventricle, it will necessarily increase the stretch, and the heart muscle says, okay, I'm just going to squeeze back that much more forcefully. And that's referred to as the Frank Starling law.
And that's good.
That's good,
Okay.
right? That's very good. Okay, so what this is going to do, like I said before, not only when you take an when you have an adrenaline rush, not only is your heart beating faster, heart rate's up, but your heart's also beating harder. It's literally squeezing harder, and that's also Frank Starling, because as you're bringing blood back faster, you're bringing more blood into your ventricles, and your heart is responding by forcefully pushing it out. So this is going to allow you to increase your ejection, right? Eject more blood. If you eject more blood, what's going on? Stroke volume's going up. And yes, in itself, that would allow the heart rate to go down. But typically, this is also happening because you're under stimulation. So your heart rate's going up as well. So if your heart rate's going up and your stroke volume's going up, that means your cardiac output's really going up, isn't it? Okay. Another thing that can influence now this is influencing ESV. Everything before that was EDV. What can influence ESV is contractility. Contractility is how forcefully the heart contracts. Contractility, as the name suggests, is how hard the heart is contracting. A greater contractility, the greater it is squeezing, the harder it's squeezing. Increased sympathetic stimulation. Right, when the bear's chasing you, the heart's beating faster and it's beating harder because contractility is going up. Epinephrine, norepinephrine, greater contractility. Thyroid hormone, greater contractility. Okay, thyroid hormone can also, if a person has low thyroid levels, they feel kind of sluggish, in part because that hormone's not influencing the heart rate. And we can reduce contractility by beta blockers. So you hear about individuals who are on beta blocker medications. Beta blockers are helping to reduce the contractility, which would also reduce the blood pressure. So beta blockers are a common medicinal pharmacological way of reducing blood pressure in patients who have high blood pressure. Okay, let me get through afterload, and then one more slide, and then I'll give you a little break. So this is also something that's going to influence stroke volume, specifically influencing the ESV, and this is afterload. Now, this also at first sounds like the amount of blood that might be in the heart after it's squeezed, right? Preload, afterload, right? That's what it sounds like. Preload was really how much the muscles are being stretched. Afterload is how much resistance there is to the heart pushing blood out. So afterload is the amount of tension necessary to squeeze the blood up and out of the ventricle. A greater afterload is going to decrease stroke volume. If it's harder, if it takes more work for my heart to push blood out, then I will push out less blood. My stroke volume will go down. Now, what will cause increased afterload? That is vasoconstriction. If your vessels are constricted, it's going to be more difficult for your heart to push blood into those restricted vessels. That's going to increase the afterload, and afterload will necessarily decrease stroke volume. Another thing is atherosclerosis. So what is atherosclerosis? Hardening of the arteries. As they harden, we saw pictures last time, and we'll see them in lab next week. As arteries harden, what happens to their lumen? It gets smaller, it narrows. Tell me about the pressure going through that tube as it narrows. Pressure's going up. If you've got atherosclerosis, you are increasing your afterload because you're making it more difficult for the heart to pump the blood into your body.
itself. It's
No,
not
afterload is
outside.
resistance to pumping out. So it's really more of an outside of the heart issue. Good. Contractility is how hard the heart is pumping. Afterload is how much resistance the heart is experiencing at the pumping. So if you have atherosclerosis, let's think about this. If you have atherosclerosis, that means that it's harder for your vessels are smaller. That means that the pressure is higher. And that means it's more difficult for the heart to pump the blood out. That means that your afterload's higher. So what's going to happen to your stroke volume? How much blood can you pump out each and every time? It's going to go down. If your stroke volume is going down, then to keep cardiac output the same, what must happen? Okay, there's another reason. We said that with age, heart rate goes up. Because with age, not that we all have atherosclerosis, but with age, most people have higher blood pressure. That higher blood pressure, again, influences afterload. It makes it more difficult for the heart to pump. So the heart has to pump more quickly to make keep the same perfusion, to keep the brain and the kidneys happen. Now, let's finish the story. What happens, though, to the heart that has to work harder? It enlarges. And when the heart enlarges, it becomes less efficient. And when it squeezes, it doesn't squeeze as hard. So what goes down? Contractility. And if you can't squeeze as hard, what's going down? Stroke volume. And if your stroke volume is going down, your heart is necessarily having to beat more often to make up for it. Okay, so it's all this, bad roller coaster, right? This big Ferris wheel that goes round and round and round. But if let's say it again, if your heart is having to work harder, if it's having to work extra hard to push the blood out, then like any muscle that has to work harder, it will get larger. It'll hypertrophy. As a heart gets larger, it's less efficient. It can't squeeze as well. You would think that a bigger heart is better. But in fact, a smaller, more compact, efficient heart is better. So as the heart enlarges, it becomes less able to squeeze well. And as a result, stroke volume goes down. As a result, the body must increase heart rate to keep cardiac output essentially the same. So that's enough up and down arrows for a couple minutes, okay? So let's think about that. Let's come back in a few minutes. So what do you think? What is afterload? How much resistance there is. Give me an example of something that increases afterload. Atherosclerosis. What is preload? How much it's stretching when it's filling, right? And what is contractility? How forcefully the heart is going to contract, okay? And all of those things are going to be influencing cardiac output. So cardiac output is going to be changing as your body needs it to change. That 6,000, Tony, that I gave you was your body at rest. Just normal resting cardiac output, okay? 6,000. That was the 75 beats per minute at 6,000 milliliters, right? 6 liters. So that 6,000 I gave you was 75 beats per minute. That's your sitting there at rest, isn't it? With a cardiac output of 6,000 and a stroke volume of 80 mils. Now, what if you had some sort of heart failure, heart damage, and the body could no longer meet the needs of your peripheral tissues? That is, it's not getting enough oxygen to your kidneys, not getting enough oxygen to your brain. Then the cardiac output would try to go up. Okay, it would try to go up. It would attempt to change it. It would try, but it's not going to be successful. It's going to try, right? So when your body can no longer maintain homeostasis, right, we call that disease. So this would be heart disease. So again, cardiac output, normal, right? Normal, average cardiac output, around 6 liters. During exercise, right, it can go up 25, 30 liters. Pretty amazing, right? It can go up 5 to 6-fold more. And under maximum, with a well-trained athlete, cardiac output can go up to 40. It can increase 6 to almost 7 times. That's going to be a combination of heart rate beating faster and stroke volume. how much blood is being pumped with each and every pump. So it's a dramatic range of what can occur with exercise, and heart failure would be anything pretty much below that. I mean, if you're not maintaining your normal 6, and if it's significantly lower than that, then your heart is somehow having difficulty keeping up with normal. Now, what is it that's influencing your heart rate? We know this. Sympathetic nervous system, increasing your heart rate, or parasympathetic, decreasing your heart rate. We got that story down. Hormones also, epinephrine specifically. We know epinephrine, norepinephrine can increase, that's sympathetic, and thyroxine. I mentioned to you in passing that thyroid hormone had an effect. So a person who's hyperthyroid, kind of describe them to me. They're like Energizer little bunnies, right? They're kind of moving around, they're burning, their heart rate's higher. A person who's hypothyroidism, they're going to have that weight gain, that sluggishness, and with that's going to come a lower heart rate. And if you're exercising, right, that is going to increase your heart rate and body temperature, okay? Heart rate increases with body temperature and lowers with a drop in body temperature. So either way, temperature and body temperature, exercise and body temperature are going to affect your heart rate. Now, there's some really nice slides like this one. This one's in 1915. And what you're going to see here is all of these things in one place. We've just talked about them individually. Here they are now in one big table. I would never, because of my own sanity, ask you to do a short answer with something like this, trying to make a concept map of all this. Although you need to be able to read through this and to appreciate all of these pieces and parts. So what can increase or influence stroke volume? Exercise. If I'm exercising more, what's happening? More blood is going back to my heart, isn't it? Now I'm having to pump more blood out to maintain my muscle, but I'm also returning more blood because my skeletal muscle pump, my diaphragm is moving more. Blood volume, right? Big changes in blood volume could change your stroke volume. If you were dehydrated, that would lower your blood pressure. If you were bleeding, same. Blood flow, changes in patterns would affect it. Sympathetic nervous system would influence it. Not only does it increase the heart rate, it also increases the contractility, how hard the heart beats, squeezes, and also hormones. Okay? So look at how all this goes together. And you will see that when venous return increases, stroke volume increases. When venous return decreases, stroke volume decreases. Now, that is going to increase or influence your EDV, your end diastolic volume. Then, the greater the contractility, the smaller the ESV. Does that make sense to you? If the heart's squeezing really hard, what will be left after it's finished squeezing? Less than if it hadn't squeezed as hard. So if the heart is really squeezing hard, high contractility, then the ESV will go down. Okay? So these are factors that influence the ESV. And together, what does this equal? EDV minus ESV equals what? Stroke volume, right? So everything on here could influence, in one way or the other, stroke volume. So let's move into EKGs. I'm going to hope that this looks really familiar to you, and that any little pieces or parts that weren't making sense to you in lab this week are now falling into place. So an EKG we know is the electrical activity of the heart's activity. We take some electrodes, we put them on the body, and we're going to see a number of electrodes, 12 in fact, in a full EKG. The computer is going to take all of that electrical activity and summate it together to give us that typical EKG wave that we've started to understand. We know this, right? P, QRS, and T wave. P wave, we know that's the atrial depolarization. There is a short latency period, so the atria will begin to contract about 25 milliseconds after the P wave starts. So the P wave, think about it, the P wave is electrical activity, isn't it? So the P wave begins, that means that the SA node is firing, and then shortly thereafter, the muscle will begin to contract. So Renee, I would call that the latency period, that little bit there for the P wave. QRS is when the ventricles are depolarizing. Again, what's causing it? The firing of the AV node. So again, there'd be the firing, you'd see the electrical activity, and then shortly thereafter, the beginning of the contraction of the ventricle. And what else is happening in the QRS that we don't see specifically, that's also the time when the atria are repolarizing. And then the T wave is the ventricular repolarization. P to R interval, talk to me. What do you remember about P to R? It's from the beginning of the P to the beginning of the QRS complex. And what does it represent? Think about the conduction system. It represents the time it takes in the internodal pathway, right? The time it takes to get from the SA node to the AV node. Do you agree? Now, what did I say was normal? 0.12 to 0.20, yes? 0.20 seconds is the same as 200 milliseconds. So it says here that if it's over 200 milliseconds, we've got a problem. Over 200 milliseconds tells me what? There's a problem in the conduction of the electrical signal going from the SA node to the AV node, and we would say that the person has a what? AV block, right? There's something blocking the signal getting to the AV node. Yes? QT, I didn't talk a lot about in lab. It was on your golden ticket page, okay? And it's going to vary a little bit. I don't have an exact number for you, but let's think about it. This is from the end of the P to R to the end of the T wave. This represents the totality of the time where the ventricles are contracting until they completely relaxed. Now, what if this is too long? If you have, quote, long QT syndrome, I didn't use that word in lab. If you have long QT syndrome, what's going on? It's taking too long for the signal to go from the AV node and down and around to the Purkinje fibers, right? There's something blocking us down there, and we might say that you have what? Not a AV block, but a bundle block. There's something blocking the signal as it travels through the bundle branches. Now, this could be all sorts of things. It could be medication. It could be a conduction problem. It could be evidence that there's been a heart attack and that damage has occurred to the muscle. So this part, let's kick back and enjoy this. We know this, right? P wave, got it. What's going on? Atria are depolarizing or systole, right? And then QRS, that is the ventricular depolarization, and hidden behind it is the atrial repolarization. And then the T wave is the ventricular repolarization. P to R, that's what you came the best at, right? And I've heard people saying, this is boring. It is. It's very repetitive. But if you do it enough times, and you think about, okay, the normal number should be between 0.12 and 0.20, then when I ask you about it on the test or the quiz, you're like, okay, I got this. So the P to R interval, right, from the beginning of the P wave to the beginning of the QRS complex. That represents the time it's taking for the signal to go from the SA node to the initiation of the AV node firing. QT, pick up right there we left off and go now to the end of the T wave. That represents the totality of time that the ventricles are squeezing and relaxing.
So why is it called the P to R interval when it's really
I know.
Q?
Isn't it lovely and confusing?
Okay.
So the P to R interval
Okay.
is not P to the R, it's not the P, beginning of the P to the top of the R. It's the beginning of the P to the beginning of the QRS.
Okay.
I know. Don't shoot me. I don't know, but you're right. Okay. Now, we learned about some arrhythmias, okay? And there were 7 arrhythmias in lab that we wanted you to look at. Do you remember what they were? Atrial flutter, atrial fibrillation, AV block, PVCs, ventricular fibrillation, tachycardia, bradycardia. Okay, I think maybe even in that order. Okay, so those are the 7 that we want you to be able to match up and think about What would that look like? Yes, I gave you a test strip on 3 of them or so in the lab on those arrhythmia worksheets. But you need to be able to think, what would be missing? If there's a missing wave, what's going on in the conduction system? I need you to make that connection. And let's go through some of these. So about 5% of the people in the country will be experiencing some abnormal heartbeats each day. So it's not unusual at all. Not all of them are a clinical problem. I mean, PVCs, premature ventricular contractions, are rather, what's the word? Meaningless. They're not a major issue. If your SA node didn't fire once, or maybe your AV node fired prematurely, right? That's what it is, PVC, right? So what does it tell you? That it just simply fired before it got the signal from the SA node. So it can happen. Little misfirings are okay. So here's the first one, though. Premature atrial contractions. Okay, this often also occurs in a healthy heart. The normal rhythm is interrupted, and this could be because of my test, right? Or caffeine, or anything that would, drugs that could increase the permeability of the SA pacemaker. That's kind of an ambiguous statement, but anything that's going to cause the SA nodes to do what? Change their ability to reach threshold, right? And normal ventricular contraction would follow that weird atrial beat. So it's just going to be like a hiccup, almost like a double heartbeat. And you may have experienced something like that. Look at this. This is where it's happening. This one was not one of your 7 in lab. Look what we have here. It's almost like there's 2 little P waves in a row, isn't there? PP, right? And the ventricle still only fired once. So that's a little premature atrial contraction. In lab, I talked about PVCs, premature ventricular contraction. But do you all see that, right? 2 P waves back to back, not that abnormal. Atrial fibrillation. Now we're talking about the atrial surface quivering up to 500 beats per minute. It is not effectively squeezing, therefore blood is not effectively going to the ventricles. If blood's not effectively going to the ventricles, then what's not happening? Your stroke volume is way decreased, and if you're squeezing 500 times a minute, you're certainly not squeezing sufficiently to do what? Push open the AV valves, right? So you got to think about anatomical and physiological consequences. So the atria are just kind of sitting there quivering. The ventricular does not follow, or maybe normal limits, because remember, the ventricles can fire on their own. Okay, so even if the SA doesn't fire, the AVs can still fire. At this point, the atria are pretty much non-functioning, and most of your blood is passively going to get into the ventricles anyway, right? Because as we said earlier, gravity helps out the atria quite a bit. So if the atria really aren't doing very well, you can get away with it because it's going to leak down anyway, and older adults have more of this. And what do you see? This is one of the ones you saw. That's kind of an irregular heartbeat with no distinctive P wave, right? Just kind of a quivering of the atria, no distinctive P wave, and I do see, even in this short little EKG, do you see a little bit of an irregularity, right, to the right? PVCs, okay, this is when the Purkinje fibers depolarize and cause the triggering of a premature contraction. Again, rather common, not that dangerous. Again, can be caused by taking my tests, right? It's a little stress, a little caffeine can cause that. And what do you see? In the lab, I showed you, it looked more like a downward spike that happened, okay? But even here, you can see something happen right here, right? And that would be a premature ventricular. You see the T wave. There's no P wave, is there? So the ventricle did what? Fired on its own, prematurely. Premature ventricular contraction. Ventricular tachycardia, VTach. This is simply what? When the heart is beating more than, or very fast, but also 4 or more PVCs without a normal heartbeat. So this could be a really serious problem. Look at this one. What do I see? A bunch of I see a bunch of QRSs. I see no P wave, right? So this would be ventricular tachycardia. This is also in addition to what we saw in lab. Ventricular fib, VFib, this is the worst of all. This is pretty much known as cardiac arrest. At this point, your ventricles are quivering, right? And if they're quivering, then nothing is getting to your brain. Basically, blood flow has stopped. We've got to get to a defibrillator at some point very, very quickly, or else this person's going to die. So the heart is basically doing nothing. Here, it looks like just a bunch of gibble. In a few seconds, what's this going to look like? Right? So that's right before the happens. Okay? That's a lovely thought. Let's stop there. Perfect timing, right? And we'll be moving into the last section. I think it's just 3 sections, into the last section on cardiac output. And that will finish up our conversation. If you're feeling good about EKGs, even better now. Feeling good about, go back and look at the ESV, EDV, the afterload, and stroke volume. And the homework and the muscle, the quiz will help you with that. Remind me, when is the muscle quiz due? Soon, right? The blood quiz was the other night. It should be coming up, right?
It's tonight. Or is it on Saturday?
I think it's coming up, but my point is, remember, there is a muscle quiz coming up. Homework first, then quiz. The heart quiz won't be due until the day before the exam. So that's not going to be until next week, but the heart quiz is going to pop up in front of this. Make sure you get that down. Feel good about that. Come back to me with any questions you have about the muscle or the heart next week. I have a handout for you, and since I already gave this out to all the Thursday hybrid lab students today, and I'm going to give it out to the 6 of you, and then I will also post this for those who are listening in who weren't here, who were sailing rather than being in class. Okay, now what this is simply a bunch of pretty blood cells, cartoons of blood cells, and you on the test will have to be able to answer all of these 6 or so questions about each of those. So make sure you can name each of these. Make sure you can tell me if it's a granulocyte or if it's an agranulocyte, if that's appropriate. How many days it survives. What's its approximate diameter in micrometers. What's its acceptable percentage. Something special about it that you can identify it or what it does. And finally, how many of those should there be in a microliter of blood. So those are all numbers. This will be a word bank kind of activity on the test. You will see the same picture, and you'll have to fill in those numbers and give me the right answer from a word bank.
You need to know the diameter.
The diameter. Now, the diameter, we know what red blood cells are. 7.5 to 8. Our lymphocytes just a little bit bigger. 10-ish. Neutrophils, eosinophils, and basophils, about 1.5 times bigger, about 12. Monocytes, up to around 20 or so, okay? 20 or 25 even, really large. So that's not so bad. Go back and look and you'll see I gave you relative sizes. So I'll see you all. Have a great weekend, and come back with any questions for me, and I'll post this again on Blackboard.